Digestive enzymes. In this video, we will cover 13 digestive enzymes produced by the human body. First, let's start by asking ourselves, what is an enzyme? An enzyme is almost always a protein, though not all proteins are enzymes. Enzymes carry out chemical reactions much faster than they would occur on their own by lowering the activation energy, the energy required for the reaction to occur. Every enzyme works most optimally at a certain temperature or temperature range, and a pH that is specific to the enzyme. For example, enzymes that catalyze reactions in the stomach work best at body temperature, about 37 degrees Celsius, and at a low pH of around 2. Most enzymes can function at lower temperatures, but they act more slowly. At too high temperatures, enzymes denature and fail to work at all. At a pH outside the optimal range, an enzyme may also deform and fail to function. Lastly, enzymes play a crucial role in our bodies, and we wouldn't be alive without them. Here is an illustration of how an enzyme works. The enzyme has a binding site in which a particular substrate can fit. Once bound, the enzyme makes it more favorable, and the chemical reaction can progress until products are formed. The products are released, and the enzymes can bind another substrate. Note the enzyme doesn't get used up during this process, so given the right conditions and enough time, enzymes can break down all the substrates available. Let's begin our journey in the mouth. Here, the extrinsic salivary glands produce two enzymes, salivary amylase and lingual lipase. Salivary amylase will act on polysaccharides, also known as starches or carbohydrates, and break them down into smaller polysaccharides. This occurs in the mouth and thus begins carbohydrate digestion. Lingual lipase is different. Although it is produced by the extrinsic salivary glands, it does not function optimally at a neutral pH. It acts minimally in the mouth and only becomes fully activated when it reaches the low pH environment of the stomach. In the stomach, it breaks lipids into molecules of glycerol and fatty acids, shown here. The two other enzymes that work optimally at a low pH are pepsin and gastric lipase. Both are secreted by chief cells of the stomach. Pepsin is technically secreted as an inactive enzyme, called pepsinogen, but is converted to pepsin, the active form, by hydrochloric acid. Pepsin then acts on polypeptides to break them into smaller polypeptides. Like lingual lipase, gastric lipase also acts on lipids and reduces them to glycerol and fatty acids. Moving on to the first part of the small intestine, the chyme or liquefied food in gastric juice encounters pancreatic enzymes secreted through the major duodenal papilla. The pancreas produces amylase, protease, and lipase. First up, pancreatic amylase. This enzyme acts on smaller polysaccharides to produce disaccharides and monosaccharides. Next, we have trypsin and chymotrypsin. These are proteases that act on smaller polypeptides to break them into dipeptides and amino acids. Lastly, the lipase produced by the pancreas is simply called pancreatic lipase. And like all other lipases we covered so far, it breaks down lipids into glycerol and fatty acid molecules. As we move distally through the small intestine, food gets churned and segmented. This is not only to mechanically mix, but also so that the chyme comes into contact with the brush border. The brush border is another name for the microvilli of the small intestine because it resembles a brush when magnified. The cells of the brush border produce enzymes that are attached rather than secreted into the lumen, so that once the enzymes break food molecules down, they can be readily absorbed by the absorptive cells of the epithelium. Let's first cover alpha-dextrinase. It breaks down a specific carbohydrate called alpha-dextrins into monosaccharides. Next, sucrase breaks down sucrose into a molecule of fructose and one molecule of glucose. Similarly, lactase breaks down lactose into one molecule of galactose and one molecule of glucose. Note that since we are pretty close to the end of our digestion journey, these end products are all simple sugars. We have a lot of brush border enzymes, so this slide contains the rest of them. Maltase, like sucrase and lactase, breaks down maltose into two glucose molecules, so more simple sugars. Our last two brush border enzymes finish off digestion of what was once proteins. Aminopeptidase acts on smaller polypeptides and produces amino acids, and dipeptidase chemically breaks dipeptides also into amino acids. It makes sense that the products of brush border enzymes are these very small molecules, because the small intestine is the last place where chemical digestion by cells of the human body occur. Whatever is not digested moves into the large intestine where bacteria break it down to feed themselves. When in balance, the gut microbiota, which includes more than just bacteria, are very beneficial and arguably crucial to the normal functioning human body. If you are interested in learning more about the gut microbiota, check out the list of published reviews on PubMed. Thank you, and I hope this video was useful.